Welcome to Healthy Minds. I'm Dr. Jeff Borenstein. Everyone is touched by psychiatric conditions, either themselves or a loved one. Do not suffer in silence. With help, there is hope. Today on Healthy Minds,
wouldn't it be great if we could treat patients before the first onset of psychosis? What if we could treat them as teenagers or children, the high-risk individuals, and prevent disease rather than just treat it?
So in addition to perhaps developing better treatments for people currently ill, your work may result in us making early detection before disease shows itself and preventing the onset of the illness. That's today on Healthy Minds. This program is brought to you in part by the American Psychiatric Association Foundation, the John and Polly Sparks Foundation, and the New York State Office of Mental Health. Welcome to Healthy Minds. I'm Dr. Jeff Borenstein. It sounds like science fiction, but scientists can now take skin cells, turn them into stem cells, which then can become brain cells. And this is allowing researchers to develop a better understanding of the brain so that we can develop better treatments. Today I speak with leading researcher, Dr. Kristen Brennan, about her work with stem cells and the brain. Kristen, thank you for joining us. I want to start off by asking you to explain, how do we learn about the brain by using stem cells?
Well, this wasn't always possible, but a discovery about 10 years ago that led to the Nobel Prize actually showed that we could take skin cells from anybody on this planet and turn those into stem cells, and those stem cells can in turn be turned into any cell type in the body. And so what that means is that I can make brain cells that are identical to those in any person, any control or any patient, and use those to begin to study disease. I think it's important to remember that it's really hard to obtain the cell types affected by disease for most diseases. Patients are very busy using their brain cells, as are controls, and so that's really limited our ability to understand what's going wrong in disease. My hope is that by using stem cells, we can recapitulate disease in a dish and really begin to see what's going on inside the brains of our patients.
So since we can't easily, and we really typically would not, get a sample of somebody's brain, it's extremely invasive, could be dangerous for the person, this gives us the next best thing of really getting a sample of their brain cells.
That's exactly right. It's with the obvious exceptions of cancer and obesity, where patients are literally paying their doctors to cut it out. For every other disease, you just cannot obtain the samples from patients. And this allows us in the laboratory to do the best alternative, to generate the cell types that are identical to those in patients and begin to study them in the lab.
We know that schizophrenia takes a number of different forms, and in reality is probably more than one illness, what we generally call schizophrenia. How does your research help us with that issue?
Well, I'd like to help us better interpret the genetic data that has been growing in the community. And so in the field at large, for years now, they have been genotyping hundreds of thousands of patients, trying to understand all of the different genetic reasons that people have schizophrenia. And so my hope is that once we better bucket schizophrenia based on genetic risk, we can model each of these different types of schizophrenia in a dish using cells that match patients from each of these smaller buckets.
So when you have these cells, what are you able to do with them? What do you learn from having the cells?
Well, the thought is that we can better understand all of the risk factors that contribute to disease. So what we're best at using stem cells is capturing disease predisposition. The cells that we make in the lab, at least today, are still really young, and they represent the cells that you or I were born with. And so we're still not great about looking at late-stage aspects of disease, but our hope is that we can contextualize risk factors that you're born with in a cell type specific manner. So for example, right now when a patient walks into the clinic, a lot of things have been going wrong in their brains for potentially a lot of years already. We know what cell types are acting aberrantly during psychosis, but we don't know if that was the first cell type to start going wrong or the 27th cell type to start going wrong. And so we can begin to unravel disease course in the dish and ask what went wrong first, what went wrong second, what went wrong third, and more importantly to ask, where can we better intervene? Is it at the first step or the second step? Wouldn't it be great if we could treat patients before the first onset of psychosis? What if we could treat them as teenagers or children, the high-risk individuals, and prevent disease rather than just treat it?
So in addition to perhaps developing better treatments for people currently ill, your work may result in us making early detection before disease shows itself and preventing the onset of the illness.
That's exactly it. That's the whole goal, is to better interpret genetic data. Since the risk factors you're born with, we can see that for the day a baby is born. We can sequence their DNA and know what they're higher risk for. So if you could understand early who was at risk for schizophrenia, you have decades to intervene with, whether it's behavioral or pharmacological interventions, and do your best to either prevent disease or minimize its effects.
I'd like you to explain a little bit about the difference between genetic and epigenetic issues, because people hear those terms and often they don't understand the difference.
These are actually really complicated terms to understand. At its most basic, genetic involves the pieces of DNA that are in every single one of your cells. The A's, the C's, the T's, the G's, the order that they're in, and the proteins that they encode. Now, each of those genes is regulated, how high they're turned on or how low they're turned off, and they can be regulated in an epigenetic factor. So there are proteins in your cells that regulate the expression of these genes, but environmental factors can also impact the expression of genes. And so, in fact, all of the products of each of our cells reflect a combination of the genetic and epigenetic factors at play.
So it's really a combination of nature, which is the genes you have, and nurture, which is the environment that those genes are living in.
Correct, and that environment is both within the cell and the environment at large outside each of us.
So your work can help look at not only the genetics, but the epigenetics, because you can see how different environmental factors can affect those cells.
Exactly, and in fact, we can do these experiments in a cleaner way in the lab. So for example, we don't all have identical twins walking around that we can abuse in any way that we want for our experimental design. So I cannot take two identical people, treat one way and one another, and ask what the impact that was. But in the lab, I can do exactly that. I can make neurons from one person, split them in half, and treat them differently. And I can exactly see how the impact of that treatment, whether that's drug exposure, whether that's inflammatory cues, how that impacts the function of the cells in the dish.
So often when we're treating someone, we may try this medicine or that medicine based upon clinical experience and the person's history. This may help us make better decisions about treatment based upon their genetic makeup.
That's exactly it. The goal is really precision medicine. I think best practice right now really seems to involve, try this drug, report back. Do you feel better or do you feel worse? But what if in the lab we could test 100 drugs, 1,000 drugs, 100,000 drugs on neurons identical to those in the patients all at once? And we could report back in 3 weeks that these are the 5 drugs that we think are best. I think you could spare patients a lot of adverse side effects by avoiding drugs that don't help them, and you could do a lot of good by getting them on drugs that do help a lot sooner in the disease.
So just as if somebody has a bacterial infection, we are able to do a test to see which antibiotic works for that bacterial infection. Perhaps we could find out which medicine would work best for this person.
That's exactly right. And we're really on the cancer model here, where You know, 20 years ago, everybody got the same chemo drugs, and some people got better and a lot of people didn't. But what you see more and more in cancer is that the doctors are genotyping tumors. They're figuring out which drugs will work best, which won't help, and targeting the right drugs to the right patients as fast as possible. And that's really what we have to get to in psychiatry too, targeting the right drugs to the right patients sooner rather than later.
I want to ask you about ethical concerns that people may have about stem cells.
When people started working with stem cells, there weren't a lot of them available to use. So whereas in the early days, embryonic stem cells were derived from extra blastocysts at the fertility clinics, today, instead, all of the stem cells in my laboratory are generated from adult patients who either we know had schizophrenia or we know didn't, and that allows us to be much more confident in our studies. We're lucky now, though, that we understand so much more about what makes a cell a stem cell that we no longer have to use embryonic cells in these studies. And in fact, I prefer not to. If we use neurons generated from an embryonic stem cell line, the truth is that we don't know anything about the person that embryo might have become. I would much rather make all of my stem cells from existing people because we have clinical histories. I'd rather they come from people that I know had schizophrenia and come from people that I know didn't have schizophrenia. And so this knowledge from decades ago has allowed us to do transformative new research beyond the early ethical concerns. The simple fact is, it is extraordinarily hard to get samples of brain from patients, and equally so from controls. The fact of the matter is we're all using our brains every single day. And this inability to access brain tissue in both health and disease has really limited our understanding of how the brain works and why it gets ill. With stem cells, we can now generate all of the cell types that comprise the brain. And we can do that across any person on the planet. And so in its most simplest forms, what that means is I can take a skin sample from any healthy control and from any patient, reprogram those into stem cells, and then direct those stem cells to become neurons. I can ask how the neurons from patients function differently than those of controls. I can begin to ask why those neurons function differently with the ultimate goal of finding ways to restore normal function, screen for new drugs and therapeutics to help patient-derived neurons act more like those in the healthy controls.
So by taking the skin sample, you can ultimately open a window into the brains of people and see what their brain cells look like.
That's the hope. We can actually take skin or blood or any cell type from patients and controls and use that to generate every cell type in the human body. And they're genetically identical to the cells that the patient was born with. In
addition to the research that you've been doing with regards to schizophrenia, you've also done research on bipolar. And I'd like you to speak a little bit about that as well.
Yeah, so the tools that we have been developing in the lab to study schizophrenia, they in fact apply easily to many other brain diseases, from bipolar to autism to Alzheimer's disease. We've looked specifically at bipolar because there's a really excellent example in bipolar treatment, lithium responsiveness. Lithium responsiveness is a really great gold standard in the sense that patients who respond when they're 20 tend to still respond when they're 80. It's a lifetime stable drug responsiveness. And more than that, that responsiveness seems to be heritable. So a patient whose father responds to lithium is himself much more likely to respond to lithium. And so for us, this is the perfect example to understand how well our stem cell-derived neurons can model known clinical responsiveness. And so the study that you're referring to, we obtained skin cells and made stem cells from lithium-responsive and non-responsive bipolar patients in order to ask if we could see differences in the way those neurons responded to lithium. And in fact, that's exactly what we saw. And so this was the first example of a time that you could correlate and even predict clinical responsiveness based on the behavior of neurons in a dish.
So in theory, a patient who's recently been diagnosed with bipolar disorder can have the skin cell sample help determine whether or not they're going to, in all likelihood, respond to lithium?
That would be the goal, yeah, exactly that. We'd like to be able to help patients get on the right drug faster and avoid taking wrong drugs that might cause unnecessary side effects.
I want to ask you about an important new technology called CRISPR. What is it and how does it work?
We've heard about CRISPR in the news a lot recently. CRISPR is a way to edit the DNA of any cell. It can be terrifying when you think about it in terms of editing our children. But in the laboratory, this is an incredibly useful tool. As I told you, we can make neurons from any patient and any control. Using CRISPR in the laboratory dish, I can then change any one or more genes to ask how the genetic risk factors that we think impact schizophrenia directly change the function of cells in the dish. So for example, when the geneticists thought there were 108, and now 145, and soon 250 risk factors linked to schizophrenia, the goal would then be in the dish to change them one at a time and in large pools to ask, do they change the function of neurons? And we can do that. We call them isogenic comparisons. So we have some neurons that we didn't change and some that we did. And so we can ask for any given risk factor what that does to the cells. We can ask if reversing them restores function, if adding extra mutations makes them worse. And it's really the most precise tool imaginable to interpret genetic risk for all disease.
So it's a way of understanding, because there are a multitude of genes that are affected, that can affect a person and increase the risk for schizophrenia. It's a way of determining which of those may have a greater risk and how.
It's about interpreting the genetic data. You might be able to look and say that patient had 17 risk factors and that patient had 72 and I have 30, and what does it mean that they have different risk factors? And so we can understand which risk factors overlap and cause additive effects, which ones are independent. We can see if some of them impact excitatory neurons and some of them impact inhibitory neurons. We can begin to understand the context of these risk factors and perhaps match them to drugs as well.
So if a particular risk factor gene is involved in producing a certain protein, we could then potentially have medications to make up for that deficit.
Exactly. Just like we talked about screening hundreds or thousands of drugs in parallel in patient-derived neurons, we can screen the impact of changing single or dozens or hundreds of genes in parallel to understand which ones are responsible or causal for disease in that particular patient.
As a scientist studying genetics, I want you to talk a little bit about the misperception that people sometimes have about themselves or about others that these illnesses are their fault.
I think there's a lot of shame around mental illness. I think that people are embarrassed about the diseases that impact them and their loved ones, and I wish as a scientist we could take that away. I remember when I was finishing my postdoc in San Diego, there was a bus shelter campaign designed to reduce the stigma around mental illness, and it was a great initiative of the city. If you think someone you love has mental illness, encourage them to see a doctor. But I always like to flip it and ask, how would that seem if we applied it to cancer? If somebody you love has cancer, encourage them to see a doctor. There are treatments available. It's so obvious when you apply it to cancer, and so important that we give the same respect and compassion to patients with mental illness. It's a different organ, but it's still disease. It's nothing that they did wrong. It's no choices that they or their loved ones make. I think it's hard enough to struggle with mental illness. If we could just let that stigma go, perhaps we could lessen the burden a little bit on patients and their families.
I think that's very well said. And the fact that science is helping us better understand the causes of these illnesses demonstrate clearly what we know to anybody else who's looking, that these illnesses aren't anybody's fault. It's the effect of their own genetic predisposition, if that exists, and environmental factors that may be beyond their control.
All of us carry genetic risk for mental illness. Some of us just happen to carry a little bit more risk or encounter a little bit more environmental stress. It's not about the choices that we make. It's unfortunately at this point still out of our control. And I really wish that we could be more compassionate to members of our world who didn't get as lucky as the rest of us.
I want to ask you how you became involved in this line of research. You're a molecular biologist, you're a basic scientist. What brought you to study this important issue?
That's a really fun question. So I'm a stem cell biologist, a developmental biologist by training. I did my PhD in the laboratory of Doug Melton at Harvard University, and it was a lab seeking to cure diabetes, using stem cells to make new beta cells, the cell type that's lost in diabetes, with the hope that we could restore their ability to regulate their sugar levels. When I finished my PhD, I thought that stem cells were an amazing tool, and it was right when the discovery that we could make stem cells from any person had been revealed. And so I immediately saw that this was going to be a game changer for disease studies, because for any disease you can think of, you could make the cell type impacted by disease. I thought the most important diseases to go after were brain diseases, because those were the ones that were inherently hardest to get tissue samples for. And so at the end of my PhD, I knew I wanted to use stem cells to model disease. I knew I wanted to look at brain disease, and I focused on the most heritable neuropsychiatric disorders that I could find, because that's what we best capture in the dish, the genetic risk. And so it turns out that if you're looking for highly heritable brain diseases, schizophrenia, bipolar, and autism top the list. And so that was why I've spent my career there. I think that they are the best brain diseases to be studying in a dish at this point in time.
In many ways, some of the most difficult illnesses to treat, which we really do need more advances to help more people.
Some of the most difficult to treat, and also, as it turns out, some of the most complex genetically. I think perhaps if I had known what we would learn in the last 10 years about schizophrenia genetics, I might have steered towards a simpler problem. This is one that will keep many of us busy for, I fear, far too long.
Where do you see the future of this research going? What do you see happening over the next 5 years, 10 years?
Well, the first thing that I see happening is all of the work by all of the geneticists is going to help us better interpret genetic risk. I hope it means that we'll be diagnosing patients more accurately, and I hope it means we're doing it earlier. We're opening the window for therapeutic intervention. It makes perfect sense that we should be treating Alzheimer's patients before their neurons die, and I think it makes equal sense that we should be treating psychiatric patients before the onset of symptoms and before the reinforcement of aberrant circuit activity. And so I hope that the first major advance is that we're diagnosing better and sooner. And then the second major advance that I think will follow in quick succession is, once we're better understanding genetic risk and diagnosing patients better, we can be drug screening better. Every current FDA-approved drug for schizophrenia has been based on the same old assay, dopamine receptor antagonism. I think we've exploited this one therapeutic angle to its limits, and it's time to find new targets for this disease and see if we can't find a broader range of drugs to help patients. And that's the other thing that I hope that we can do in the lab, is unbiased screens, independent of dopamine signaling, to find new avenues of treatment.
So what you would do is take these samples of brain cells and try different potential medications and see the effect upon those cells.
Yeah, the biggest vision, of course, would be take a patient, get their skin cells, turn those into stem cells and brain cells, and do a personalized screen on every single patient. That, of course, is a huge undertaking, and so rather than that, if the geneticist could break schizophrenia down into 12 or 200 or 800 different types of schizophrenia, do those 800 drug screens, test 100,000 drugs for each type of schizophrenia once genetically binned. I think even then you're getting towards a more precise form of medicine.
Kristen, I want to thank you for the work that you've been doing, for the work that you're going to continue to do, and thank you for joining us here today.
It was my absolute pleasure. Thank you so much.
Researchers are developing new techniques to study the brain so we can develop new treatments, methods of preventions, and cures for psychiatric conditions. These will help countless people who haven't been helped before. Remember, with help, there is hope. Until next time, I'm Dr. Jeff Borenstein. Do not suffer in silence. With help, there is hope. This program is brought to you in part by the American Psychiatric Association Foundation, the John and Polly Sparks Foundation, and the New York State Office of Mental Health. Remember, with help, there is hope. Until next time, I'm Dr. Jeff Borenstein.